Everyone, welcome to the video lecture for Unit 4, Module 8.3. In this one, we're going to highlight reabsorption and secretion. As always, here are your learning objectives. Please be sure to refer to these when studying for both your lecture quiz as well as the unit exam. All right, let's go ahead and get started. Reabsorption recovers useful materials that have entered the filtrate. Secretion ejects waste, toxins, or other undesirable solutes that did not leave the bloodstream at the glomerulus. Both processes take place in every segment of the nephron except for the renal corpuscle. Their relative importance changes from segment to segment. The cells of the proximal convoluted tubule normally reabsorb about 60 to 70% of the volume of the filtrate produced in the renal corpuscle. The reabsorbed materials enter the peritubular fluid, diffuse into the peritubular capillaries, and are quickly returned to the circulation. The PCT has 5 major functions. The first is reabsorption of organic nutrients. Under normal circumstances, before the tubular fluid enters the nephron loop, the PCT reabsorbs more than 90% of the glucose, amino acids, and other organic nutrients in the fluid. This reabsorption involves a combination of facilitated transport and cotransport. 2 is active reabsorption of ions. The PCT actively transports several ions, including sodium, potassium, and bicarbonate ions, plus magnesium, phosphate, and sulfate ions. Although the ion pumps involved are individually regulated, they may be influenced by circulating ion or hormone levels. For example, angiotensin II stimulates sodium reabsorption along the PCT. By absorbing carbon dioxide, the PCT indirectly recaptures about 90% of the bicarbonate ions from tubular fluid. Bicarbonate is important in stabilizing blood pH. Number 3 is the reabsorption of water. The reabsorptive processes have a direct effect on the solute concentrations inside and outside the tubules. The filtrate entering the PCT has the same osmotic concentration as that of the surrounding peritubular fluid. As reabsorption proceeds, the solute concentration of tubular fluid decreases, and that of the peritubular fluid and adjacent capillaries increases. Osmosis then pulls water out of the tubular fluid and into the peritubular fluid. Along the PCT, this mechanism results in the reabsorption of about 108 liters of water each day. Number 4 is passive reabsorption of ions. As active reabsorption of ions takes place and water leaves the tubular fluid by osmosis, the concentration of other solutes in the tubular fluid increases above that in the peritubular fluid. If the tubular cells are permeable to them, those solutes move across the tubular cells and into the peritubular fluid by passive diffusion. Urea, chloride ions, and lipid-soluble materials may diffuse out of the PCT in this way. Such diffusion further decreases the solute concentration of the tubular fluid and promotes additional water reabsorption by osmosis. And 5 is secretion. Active secretion of hydrogen ions takes place along the PCT. You can see this in Table 26-4. Because the PCT and the DCT secrete similar substances, the DCT carries out comparatively little reabsorption. We'll cover secretory mechanisms when we discuss the DCT. Sodium ion reabsorption plays an important role in all of these processes. Sodium ions may enter the tubular cells by diffusion through the sodium leak channels, by the sodium-linked cotransport of glucose, amino acids, or other organic solutes, or by countertransport for hydrogen ions. Once inside the tubular cells, sodium ions diffuse towards the basement membrane. The plasma membrane in this area contains sodium-potassium exchange pumps that eject sodium ions in exchange for extracellular potassium ions. Reabsorbed sodium ions then diffuse to the peritubular fluid and into adjacent peritubular capillaries. The reabsorption of ions and compounds along the PCT involves many carrier proteins. Some people have an inherited inability to manufacture one or more of these carrier proteins, and for this reason, these individuals are unable to recover specific solutes from tubular fluid. In renal glycosuria, for example, a defective glucose carrier protein makes it impossible for the PCT to reabsorb glucose from tubular fluid. Approximately 60 to 70% of the volume of the filtrate produced by the glomerulus is being reabsorbed before the tubular fluid reaches the nephron loop. In the process, useful organic substrates and many mineral ions have been reclaimed. The nephron loop reabsorbs about half of the remaining water and 2/3 of the remaining sodium and chloride ions. This reabsorption takes place efficiently according to the principle of countercurrent exchange. The thin descending limb and the thick ascending limb of the nephron loop lie very close together. They are separated only by peritubular fluid. The exchange between these segments is called countercurrent multiplication. Countercurrent refers to the fact that the exchange takes place between the fluids moving in opposite directions. Tubular fluid in the descending limb flows towards the renal pelvis, while tubular fluid in the ascending limb flows towards the cortex. Multiplication refers to the fact that the effect of the exchange increases as movement of the fluid continues. The 2 parallel limbs of the nephron loop have very different permeability characteristics. The thin descending limb is permeable to water, but relatively impermeable to solutes. The thick ascending limb is relatively impermeable to both water and solutes, but it contains active transport mechanisms that pump sodium and chloride ions from the tubular fluid into the peritubular fluid of the medulla. A quick overview of countercurrent multiplication will help you make sense of the details. Sodium and chloride ions are pumped out of the thick ascending limb and into the peritubular fluid. This dilutes the tubular fluid. The pumping action increases the osmotic concentration in the peritubular fluid around the descending limb. This creates a small concentration difference between the tubular fluid and the peritubular fluid in the renal medulla. The concentration difference results in an osmotic flow of water out of the thin descending limb and into the peritubular fluid. As a result, the solute concentration increases in the thin descending limb. The arrival of the highly concentrated solution in the thick ascending limb speeds up the transport of sodium and chloride ions into the peritubular fluid. Solute pumping at the ascending limb leads to higher solute concentrations in the descending limb, which then brings about the increased solute pumping in the ascending limb. Notice that this process is a simple positive feedback loop that multiplies the concentration difference between the hypotonic tubular fluid in the ascending limb and the hypertonic peritubular fluid in the renal medulla. We can now take a closer look at the mechanics of the process. The figure here diagrams ion transport across the epithelium of the thick ascending limb. Active transport at the apical surface moves sodium, potassium, and chloride ions out of the tubular fluid. The carrier is called the sodium-potassium Cl transporter because each cycle of the pump carries a sodium ion, a potassium ion, and 2 chloride ions into the cell. Then, co-transport carriers pump potassium and chloride ions into the peritubular fluid. However, potassium ions are removed from the peritubular fluid as the sodium-potassium exchange pump pumps sodium ions out of the tubular cell. The potassium ions then diffuse back into the lumen of the tubule through the potassium leak channels. The net result is that sodium and chloride enter the peritubular fluid of the renal medulla. The removal of sodium and chloride ions from the tubular fluid in the ascending limb raises the osmotic concentration of the peritubular fluid around the descending limb. Recall that the thin descending limb is permeable to water but not to solutes. As the tubular fluid travels deeper into the medulla within the thin descending limb, osmosis moves water into the peritubular fluid. Solutes remain behind. As a result, tubular fluid at the turn of the nephron loop has a higher osmotic concentration than it did at the start. The pumping mechanism of the thick ascending limb is highly effective. Almost 2/3 of the sodium and chloride ions that enter it are pumped out of the tubular fluid before that fluid reaches the DCT. In other tissues, differences in solute concentrations are quickly resolved by osmosis. However, osmosis cannot take place across the wall of the thick ascending limb because the epithelium there is impermeable to water. So, as sodium and chloride are removed, the solute concentration in the tubular fluid decreases. Tubular fluid arrives at the DCT with an osmotic concentration of only about 100 milliosmoles per liter. This value is 1/3 the concentration of the peritubular fluid of the renal cortex. The rate of ion transport across the thick ascending limb is proportional to an ion's concentration in tubular fluid. As a result, more sodium and chloride ions are pumped into the medulla at the start of the thick ascending limb, where sodium chloride concentrations are highest, then near the cortex. This regional difference in the rate of ion transport is the basis of the concentration gradient within the medulla. Normally, the maximum solute concentration of the peritubular fluid near the turn of the nephron loop is about 1200 milliosmoles per liter. Sodium and chloride ions pumped out of the loop's ascending limb make up about 2/3 of that gradient, about 750 milliosmoles. The rest of the concentration gradient results from the presence of urea. To understand how urea arrives at the medulla, let's look ahead to the events in the last segments of the collecting system. The thick ascending limb of the nephron loop, the DCT, and the collecting ducts are all impermeable to urea. As water is reabsorbed, the concentration of urea gradually increases in the tubular fluid. When the tubular fluid reaches the papillary duct, it typically contains urea at a concentration of about 400 milliosmoles per liter. Because the papillary ducts are permeable to urea, the concentration in the deepest parts of the medulla also averages 450. The countercurrent mechanism performs 2 functions. 1. It effectively reabsorbs solutes and water before the tubular fluid reaches the DCT of the collecting system. 2. It establishes a concentration gradient that permits passive reabsorption of water from the tubular fluid in the collecting system. Circulating levels of antidiuretic hormone, or ADH, regulate this reabsorption. In summary, countercurrent multiplication is a way to either concentrate or dilute urine. The tubular fluid entering the descending limb of the nephron loop has an osmotic concentration of roughly 300 milliosmoles per liter, due primarily to the presence of ions such as sodium and chloride. The concentration of organic waste, such as urea, is low. About half the tubular fluid entering the nephron loop is then reabsorbed along the thin descending limb. 2/3 of the sodium and chloride is reabsorbed through the thick ascending limb. As a result, the DCT receives a reduced volume of tubular fluid with an osmotic concentration of about 100 milliosmoles per liter. Urea and other organic waste, which were not pumped out of the thick ascending limb, now represent a significant proportion of the dissolved solutes. As we've just seen, the composition and volume of tubular fluid changes dramatically as it flows from the capsular space to the distal convoluted tubule. Only 15 to 20% of the initial filtrate volume reaches the DCT. The concentration of electrolytes and organic waste in the arriving tubular fluid no longer resemble the concentrations in blood plasma. Selective reabsorption or secretion, primarily along the DCT, makes the final adjustments in the solute composition and the volume of the tubular fluid. Throughout most of the DCT, the tubular cells actively transport sodium and chloride out of the tubular fluid. Tubular cells along the distal portions of the DCT also contain ion pumps that reabsorb tubular sodium in exchange for another cation, usually potassium. The hormone aldosterone, produced by the adrenal cortex, controls the sodium channels and the ion pump. This hormone stimulates the synthesis and incorporation of sodium channels and sodium ion pumps in the plasma membranes along the DCT and collecting duct. The net result is a reduction in the number of sodium ions that are lost in urine. However, sodium ion conservation is associated with potassium ion loss. Prolonged aldosterone stimulation can therefore produce hypoalkemia, which is a dangerous reduction in the plasma potassium levels. The secretion of aldosterone and its actions on the DCT and collecting system are opposed by the natriuretic peptides ANP and BNP. The DCT is also the primary site of calcium reabsorption. Circulating levels of parathyroid hormone and calcitriol regulate this process. The blood entering the peritubular capillaries still contains a number of potentially undesirable substances that did not cross the filtration membrane at the glomerulus. In most cases, the concentration of these substances are too low to cause physiological problems. However, any ions or compounds in the peritubular capillaries will diffuse into the peritubular fluid. If those concentrations become too high, the tubular cells may absorb these substances from the peritubular fluid and secrete them into the tubular fluid. Table 26-3, which you'll see soon, lists some of the substances secreted into tubular fluid by the proximal and distal convoluted tubules. The rate of potassium and hydrogen secretion increases or decreases in response to changes in their concentrations in peritubular fluid. The higher their concentration in the peritubular fluid, the higher the rate of secretion. Potassium and hydrogen ions merit special attention because their concentrations in body fluids must be maintained within narrow limits. The figure here diagrams the mechanisms of potassium secretion. In effect, tubular cells trade sodium ions in the tubular fluid for excess potassium ions in body fluids. Potassium ions are removed from the peritubular fluid in exchange for sodium ions from the tubular fluid. These potassium ions diffuse into the lumen of the DCT through potassium leak channels at the apical surface of the tubular cells. Hydrogen ion secretion is also associated with the reabsorption of sodium. The figure here, or that you'll see on the next slide, depicts two routes of secretion. Both involve the generation of carbonic acid by the enzyme carbonic anhydrase. Hydrogen ions generated by the dissociation of carbonic acid are secreted by sodium-linked countertransport in exchange for sodium in the tubular fluid. The bicarbonate ions diffuse into the peritubular fluid and then into the bloodstream. There, they help prevent changes in plasma pH. Hydrogen ion secretion acidifies the tubular fluid while increasing the pH of the blood. Hydrogen ion secretion speeds up when the pH of the blood decreases. This can happen in lactic acidosis, which can develop after exhaustive muscle activity, or in ketoacidosis, which can develop in starvation or diabetes mellitus. The combination of hydrogen removal and bicarbonate production by the kidneys plays an important role in the control of blood pH. Because one of the secretory pathways is aldosterone sensitive, aldosterone stimulates hydrogen secretion. Prolonged aldosterone stimulation can cause alkalosis, or abnormally high blood pH. Note that the production of lactic acid and ketone bodies during the post-absorptive state could cause acidosis. Under these conditions, the PCT and the DCT deaminate amino acids in reactions that strip off the amino groups, or the NH2 group. The reaction sequence ties up hydrogen and yields both ammonium ions, or NH4, and bicarbonate. The ammonium ions are then pumped into the tubular fluid by sodium-linked countertransport, and the bicarbonate ions enter the bloodstream by way of the peritubular fluid. Tubular deamination thus has two major benefits. It provides carbon change suitable for catabolism. It also generates bicarbonate ions that add to the buffering capacity of the plasma. The collecting ducts receive tubular fluid from many nephrons and carry it towards the renal sinus through the concentration gradient in the medulla. The normal amount of water and solute loss in the collecting system is regulated in two ways: by aldosterone, which controls sodium ion pumps along most of the DCT and the proximal portion of the collecting system. As we've noted, these actions are opposed by natriuretic peptides, and by ADH, which controls the permeability of the DCT and the collecting system to water. The secretion of ADH is suppressed by natriuretic peptides, and this, combined with the effects of the natriuretic peptide on aldosterone secretion and action, can dramatically increase urinary water losses. The collecting system also has other reabsorptive and secretory functions. Most of them are important in the control of body fluid pH. The collecting system reabsorbs sodium ions, bicarbonate ions, and urea as follows. When discussing sodium ion reabsorption, the collecting system contains aldosterone-sensitive ion pumps that exchange sodium in tubular fluid for potassium in peritubular fluid. Bicarbonate ions are reabsorbed in exchange for chloride ions in the peritubular fluid. Speaking on urea reabsorption, the concentration of urea in the tubular fluid entering the collecting duct is relatively high. The fluid entering the papillary duct generally has the same osmotic concentration as that of the interstitial fluid of the medulla, about 1200 milliosmoles per liter, but contains a much higher concentration of urea. As a result, urea tends to diffuse out of the tubular fluid and into the peritubular fluid in the deepest portion of the medulla. The collecting system is important in controlling the pH of body fluids through the secretion of hydrogen or bicarbonate ions. If the pH of the peritubular fluid decreases, carrier proteins pump hydrogen ions into the tubular fluid and reabsorb bicarbonate ions that help restore normal pH. If the pH of the peritubular fluid rises, a much less common event, the collecting system secretes bicarbonate ions and pumps hydrogen ions into the tubular fluid. The net result is that the body eliminates a buffer and gains hydrogen ions that lower the pH. Urine volume and osmotic concentration are regulated through the control of water reabsorption. Water is reabsorbed by osmosis along the proximal convoluted tubule and the descending limb of the nephron loop. The water permeabilities of these regions cannot be adjusted. As a result, water reabsorption takes place wherever the osmotic concentration of the peritubular fluid is greater than that of the tubular fluid. The ascending limb of the nephron loop is impermeable to water. In the distal convoluted tubule and the collecting systems, 1 to 2% of the volume of water in the original filtrate is recovered during sodium ion reabsorption. All these water movements represent obligatory water reabsorption because they cannot be prevented. This reabsorption usually recovers about 85% of the volume of the filtrate. The volume of water lost in urine depends on how much of the remaining water in the tubular fluid is reabsorbed along the DCT in the collecting system. This remaining water represents about 15% of the filtrate volume, or approximately 27 liters per day. The amount reabsorbed can be precisely controlled by a process called facultative water reabsorption. Precise control is possible because these segments are relatively impermeable to water except in the presence of ADH. This hormone causes special water channels, or aquaporins, to be inserted into the apical plasma membranes. These water channels dramatically enhance the rate of osmotic water movement. The higher the circulating levels of ADH, the greater the number of water channels and the greater the water permeability of these segments. As noted earlier in this module, the tubular fluid arriving at the DCT has an osmotic concentration of only about 100 milliosmoles per liter. In the presence of ADH, osmosis takes place. Water moves out of the DCT into the osmotic concentration of the tubular fluid equals that of the surrounding cortex, roughly 300 milliosmoles per liter. The tubular fluid then flows along the collecting duct, which passes through the concentration gradient of the medulla. Additional water is then reabsorbed. The urine reaching the minor calyx has an osmotic concentration closer to 1200 milliosmoles per liter. Just how closely the osmotic concentration approaches 1200 milliosmoles per liter depends on how much ADH is present. Without ADH, water is not reabsorbed in these segments, so all the fluid reaching the DCT is lost in the urine. The person then produces large amounts of very dilute urine. This is just what happens in the cases of diabetes insipidus. With this condition, urinary water losses may reach 24 liters per day, and the urine osmotic concentration is about 30 to 400 milliosmoles per liter. As ADH levels rise, the DCT and collecting system become more permeable to water. As a result, the amount of water reabsorbed increases. At the same time, the osmotic concentration of the urine climbs. Under maximum ADH stimulation, the DCT and collecting system becomes so permeable to water that the osmotic concentration of the urine equals that of the deepest portion of the medulla. Note that the concentration of urine can never exceed that of the medulla because the concentrating mechanism relies on osmosis. The hypothalamus continually secretes ADH at low levels. For this reason, the DCT and collecting system always have a significant degree of water permeability. At these low ADH levels, the DCT reabsorbs approximately 9 liters of water per day, or about 5% of the original volume of the filtrate produced by the glomeruli. At normal ADH levels, the collecting system reabsorbs about 16.8 liters per day, or about 9.3% of the original volume of the filtrate. A healthy adult typically produces about 1,200 milliliters of urine per day. Its osmotic concentration range is roughly 855 to 1,355 milliosmoles per liter. The effects of ADH are opposed by those of the natriuretic peptides A and B and BNP. These hormones stimulate the production of a large volume of relatively dilute urine. This water loss reduces plasma volume to normal. The solutes of water reabsorbed in the renal medulla must be returned to the bloodstream without disrupting the concentration gradient. This return is the function of the vasa recta. Recall that the vasa recta are long, straight capillaries that parallel the long nephron loop of the juxtamedullary nephrons. Blood entering the vasa recta from the peritubular capillaries has an osmotic concentration of approximately 300 milliosmoles per liter. As the blood descends into the medulla, it gradually increases in osmotic concentration as the solute concentration in the peritubular fluid increases. This increase in blood osmotic concentration involves both solute absorption and water loss. Solute absorption predominates, however, because the plasma protein limit the osmotic flow of water out of the blood. Blood ascending towards the cortex gradually decreases in osmotic concentration as the solute concentration of the peritubular fluid decreases. Again, this decrease involves both solute diffusion and osmosis. In this case, osmosis predominates because the presence of plasma protein does not oppose the osmotic flow of water into the blood. The net results are that 1. Some of the solutes are absorbed in the descending portion of the vasa recta, do not diffuse out of the ascending portion, and 2. More water moves into the ascending portion of the vasa recta than moves out of the descending portion. Thus, the vasa recta carries both water and solutes out of the medulla. Under normal conditions, the removal of solutes and water by the vasa recta precisely balances the rate of solute reabsorption and osmosis in the medulla. As we've seen, more than 99% of the 180 liters of filtrate produced each day by the glomeruli is reabsorbed. It never reaches the renal pelvis for elimination. General characteristics of the remaining filtrate, or normal urine, are listed in Table 26-5. However, the composition of urine produced each day varies with the metabolic and hormonal events underway. The composition and concentration of urine are two related but distinct properties. The composition of urine reflects the filtration, reabsorption, and secretion activities of the nephrons. Some substances, such as urea, are neither actively excreted nor reabsorbed along the nephron. In contrast, organic nutrients are completely reabsorbed. Other substances, such as creatinine, are missed by filtration but are actively secreted into the tubular fluid. Filtration, reabsorption, and secretion determine the kinds and amounts of substances excreted in urine. The concentration of these substances in a given urine sample depends on the osmotic movement of water across the walls of the tubules and collecting ducts. Because the composition and concentration of urine vary independently, you can produce a small volume of concentrated urine or a large volume of dilute urine and still excrete the same amount of dissolved substances. For this reason, physicians who are interested in a detailed assessment of renal function commonly analyze the urine produced over a 24-hour period rather than a single urine sample. Urinalysis is the analysis of a urine sample. It's an important diagnostic tool, even in high-technology medicine. A standard urinalysis includes an assessment of the color and appearance of urine. These two characteristics can be determined without specialized equipment. In the 17th century, physicians often classified the taste of urine as sweet, salty, and so on, but thankfully, quantitative and analytical tests have long since replaced the taste bud assay. Normal urine is a clear, sterile solution. Its yellow color comes from the pigment urobilin. The kidneys generate this pigment from the urobilinogens produced by the intestinal bacteria and absorbed in the colon. The characteristic odor of urine is due to the evaporation of small molecules such as ammonia. Other substances not normally present, such as acetone or other ketone bodies, can also impart a distinctive smell. Table 26-6 gives some typical values obtained from urinalysis. And on slides 26 and 27, it just highlights the 7 steps of renal function.